Hello again. So we come to the next part, translation and the structure of proteins. Translation is the production of proteins using mRNA as a template, and the genetic code on mRNA is in the form of base triplets called codons. An important part of the translation process are tRNA molecules or transfer RNA molecules. These are found in the cytoplasm and fold back on themselves due to complementary base pairing, but they are in fact of course made of single strands of RNA. Here we see a single tRNA molecule with its amino acid attachment site there, and this particular amino acid that it's carrying is glutamic acid. It has three bases exposed, and these bases comprise the anticodon, which is there, and of course the anticodon is complementary to the codon on the mRNA strand. And each tRNA molecule is specific to a certain amino acid. Another important part of the process are ribosomes. They have a binding site for mRNA and three binding sites for tRNA molecules. Site P holds the tRNA carrying the growing polypeptide chain. Site A receives a tRNA bearing an amino acid to be joined to the growing polypeptide by a peptide bond. And site E discharges a tRNA molecule from the ribosome once its amino acid has become part of the chain. Now, you do not need to know this detail, but it helps to visualize the process of translation itself. So if we have a look at the process of translation, firstly a ribosome attaches to an mRNA molecule, then a tRNA bearing the first amino acid, methionine, arrives at site P of the ribosome, its anticodon being complementary to the first codon on the mRNA strand. Then another tRNA bearing the next amino acid arrives at the next codon at site A of the ribosome. Then a peptide bond forms between the two amino acids. Then the ribosome moves along by one codon. The tRNA molecule bearing methionine found itself then on site E of the ribosome and so was discharged. Another tRNA molecule bearing another amino acid arrives at site A. Once again, another peptide bond forms. The ribosome then moves along by another codon. And once again, the tRNA finding itself on site E of the ribosome is discharged. This process continues until the ribosomes moved its way along the entire mature mRNA transcript until the complete polypeptide chain is formed. All those amino acids joined together by peptide bonds. More than one ribosome at a time may be attached to an mRNA molecule, so multiple polypeptides can be produced. Such a string of ribosomes is called a polyribosome. The human genome contains about 30,000 genes, but there are between about 500,000 to 1 million human proteins produced. This is due to alternative splicing. This is where the primary mRNA transcript is spliced in several ways to produce different mature mRNA transcripts. So, for example, if this gene has exons 1, 2 and 3, this mature mRNA transcript will have exons 2 and 3. This one has 1 and 2, and this final variant has exons 1 and 3. Different arrangements of exons, but they are all in the same order as they're found in the original gene. Now, if we look at the structure and function of proteins, you know that the DNA base sequence determines amino acid sequences in polypeptides. Amino acid sequence determines the protein's structure and function, and that genotype through gene expression plus various environmental factors can influence the phenotype of the organism. Now, if we have a look at this chain of amino acids strung together after translation, intermolecular forces such as hydrogen bonds and other molecular links between amino acids such as sulfur bridges determine the final 3D structure of the protein and hence its function. So in terms of the final protein structures that we can get, we can have strands of polypeptide arranged in a parallel formation, such as collagen. You can have roughly spherical polypeptides like enzymes, and you can have other roughly spherical polypeptides incorporating another inorganic element like iron, for example, to make hemoglobin that we find in what's called conjugated proteins. So finally, if we look at the functions of some different proteins, firstly we have enzymes. These are made of protein folded in a way which exposes an active site which combines with a specific substrate. They speed up important cellular biochemical processes like respiration, photosynthesis, and protein synthesis. Next we have structural proteins. These are one of the components of cell membranes and the membranes of cell organelles, and they play a vital role in every single cell. Next we have hormones. These have a regulatory effect on growth and metabolism. They're made by endocrine glands in the body, and they are chemical messengers which travel in the blood to target organs. For example, insulin, antidiuretic hormone, and human growth hormone. Finally, we have antibodies, and these are Y-shaped proteins made by the white blood cells to help defend the body against diseases. Okay, thank you very much once again.